Hello everyone. Now let's discuss on a few techniques like ELISA and RIA. First question, RIA, radio immunoassay for 3 marks. So you first give the definition of RIA, radio immunoassay. Next we can move on to the principle. You write about the principle, that is it uses an immune reactions, antigen antibody reactions to estimate the ligand. Here the antigen and radioactive antigen and antibody is made to react to form the complex, and the unbound antigen and radioactive antigen, unbound, which is unbound, is washed away and the radioactivity is being measured, and the concentration of the radioactivity gives the amount of ligand concentration, that is the antigen antibody concentration. So, the RIA, radio immunoassay advantages are like it is highly specific and sensitive. Then it has a disadvantages like it is since it is using radioactive substance, there is radiation hazard and it requires special trained persons and special license because it is all handling radioactive materials. And also since it is radioactive substance, the arrangements is also quite expensive. Next we will see about the procedure. Next give a brief discussion on the procedure. Add a known amount of the test sample and the labeled antigen, radioactive labeled antigen, to the micro well. Then it is incubated for certain time, and during the incubation there is reaction taking place between the antigen and antibody. And next is the washing procedure, that is to remove the unbound antigens, and radioactivity is measured by the counters. Radioactive activity will give the amount, intensity of the radioactivity is proportional or related with the concentration of antigens in the test sample. One of the main advantages of this is it is very sensitive to even a lower concentration of antigens. Next is the application. It is used for the application of analysis of hormones, vitamins, metabolites, diagnostic markers like ACTH, FSH, T3, T4, glucagon, insulin, testosterone, vitamin B12, prostaglandin, glucocorticoids, etc. All this estimation of these hormones and vitamins. Then other diagnostic markers also. Then it is used for therapeutic drug monitoring. Drugs like morphines, etc. is being monitored with the RIA techniques. Next is for diagnostic purposes. In diagnosis of HIV, hepatitis, etc. it is used. This application alone can be asked. For lesser marks it can be asked for the applications alone can be asked. So, you can, this applications is very important. Next question is ELISA. ELISA 3 marks question. So, first you can give the definition what is ELISA means, enzyme linked immunosorbent assay, and you give a brief description. It is a biochemical technique used mainly in immunology to detect the presence of any antigen or antibody in the sample, and it is using the principle of immune reactions like in RIA, antigen antibody reactions. It differs from RIA in detection method. The detection method is mainly based upon enzyme catalyzed reaction or fluorescent probe, and it is not depending upon radioactivity. ELISA application mainly it is used for hormone measurements, growth factors, tumor markers, bacterial and viral antigen antibody test against microbes, antigens or antibodies in biological fluids. So, it is used in assays of hormones and growth markers and also tumor markers. Then it is used also for detection of various bacterial and viral antigens or antibodies. Now, you need to explain what are the different types of ELISAs. So, first you explain what is the definition and also principle, that is antigen antibody immune response, antigen antibody reaction is being measured. Next is the different types. Non-competitive and competitive. Non-competitive assay, otherwise known as sandwich method, it is divided into two, that is antigen measuring and antibody measuring. Antigen measuring means the wells will be coated with antibodies and we will be providing antigens. In antibody, the well will be coated with antigens and enzyme labeled antibodies is being provided. In competitive binding assays, the titer will be coated with antibodies and enzyme labeled antigens. Next one is competitive ELISA. The labeled antigen in competitive ELISA, these labeled antigens competes for a primary antibody binding sites with the sample antigen which is unlabeled. The more antigen in the samples means the less labeled antigen is retained in the well, and the weaker will be the signal provided. Next ELISA technique is sandwich ELISA. The ELISA plate is coated with antibody to detect specific antigen. Next terminology in ELISA is indirect ELISA. In indirect ELISA, you can give a brief description what it is, how it is done, what are the steps to be followed. The protein antigen to be tested is added to each well of ELISA plate, where it is given time for it to bind with the plastic well, and then a solution of non-reacting proteins is added to block the plastic surface in the well that remains uncoated. Then that is the preparation of the well. So how it is prepared, how it is coated with antigen, and then we are adding the serum which contains a mixture of antibodies with unknown concentration, and antigen antibody reaction is being taken place. After that a secondary antibody is also added. That's why it is called as indirect method. A second antibody is added which binds to the antibody to bound to the test antigen in the well, and the secondary antibody is usually an enzyme, has an enzyme attached to it. A substrate of these enzymes is then added. Often this substrate changes the color upon reactions with the antibodies, which is bound to the primary antibodies, which strongly implies that the donor has an immune reactions to the test antigens. The higher the concentration of the primary antibody that was present in the serum, the stronger will be the color change, and we are measuring the color intensity. Next is non-competitive, that is divided into two, antigen and antibody. In that antigen measuring system, the titer well is coated with suitable antibodies, and patient sample is added which is containing the antigens. It is incubated for certain time so that the antigen antibody reaction is completed, and next step is washing to remove the unbound antigen, and this next we are adding antibody labeled with the enzyme, then that is incubated till the antigen binds labeled to the labeled antibody, and then we are washing it to remove the unbound labeled antibody, and substrate is also added and incubated, and we are measuring the color intensity. Same way antibody measuring system, just the difference is that we are adding we are using a well which is coated with suitable antigens. For antibody measuring we are using antigen coated, and patient sample with antibodies added, then incubated and washing, then an antibody labeled with enzyme is being added, and it is incubated till the labeled antibody binds to the antigen antibody complex, and then it is washed to remove the unbound, and then substrate is added, incubated to get a colored solution, and the color intensity is measured. Advantages of ELISA: it is sensitive, it is reproducible, minimal reagents, quantitative and qualitative experiments can be done. Then it is greater scopes because the wells can be coated with antigen or antibodies. Then it is also suited for automation, fully automated machines can operate the ELISA. Then it has no radiations or hazards as that of radioimmunoassay. Then applications, same as RIA, that is for diagnosis, for the estimation of hormones, vitamins, then diagnostic markers, then for therapeutic drugs, then also for detection of infections, etc. So application alone can be asked, or types of ELISA can be asked for lesser marks. So you can answer accordingly. If types of ELISA types alone is asked, you can explain different procedures and different types and how it is done, a brief description of how it is done, how it is differing from each other. Then applications alone can be asked. Thank you.